So my name is Qin Liu. I'm a postdoc at Dr. Petra Reeder's lab at the brain simulation section. Today, I and my colleagues, Leon and Valerie, we're going to present our work in modeling the mechanisms of consciousness and memory. So first, I'd like to quote Richard Feynman. Uh, he once said, "What I cannot create, I do not understand." So this is actually my very first motivation that motivated me to join the field of computational neuroscience. So many years ago, I was in the field of physics, and I also studied in an experiment neurology lab. So although at that point we collected a lot of data, I saw all those experimentalists performed very fantastic research in neuroscience. I still feel quite insecure because I always thought like what they found was only part of the story, and I wanted to know the whole story behind all those findings. And then I realized what I wanted to do actually is just to create a model that can perform the brain functions from the bottom to the top. But brain functions are very complex. What exactly, um, is that does the simulating a brain function means academically? So, so here is one famous conceptual art, One and the Three Chairs. So in this piece of art, the artist Joseph Kosuth, he presented a chair, a real chair, and a picture of that real chair, and then a dictionary entry of the word chair. So I guess the artist point was, the concept chair exists in multiple dimensions in different semantic fields. But this is also a very good example of neuroscience problem. So whenever we perform a very simple activity, for example, just looking at that chair, we actually utilized a lot of complex brain functions. So first of all, we need recognition. We saw that chair, and then we need to recognize, okay, this is the chair. And then we utilize the function of categorization. So we know obviously it's a chair, it's in the category of chair. And we also know what type of chair it is. Is it a big one? Is it a new one? So we performed categorization already. And then we also performed audio-visual association. So because we know the word chair in English or in other languages, so maybe we didn't even realize we did this, but our brain obviously is doing this all the time. So this is the so-called naming, and this is the building blocks of reading and language. And also we performed short-term memory as well, because we saw the chair, the image of the chair is maintained in our short-term working memory. And then we also performed long-term memory retrieval because we know, have we seen this chair before? Or is this a new one? Or is it similar to other chairs we saw before that are stored in our long-term memory? So although this is a very simplistic movement, we simply saw a chair, but our brain did a lot of work by utilizing a lot of complex brain functions. We can roughly rank our brain functions from lower level functions to higher level functions. Here, lower level functions generally refer to those ones who appear early in the evolutionary lines, and higher level functions appeared later along the evolutionary line. So for those lower level functions, they generally tend to be localized in some brain regions, for example, like visual functions or motor functions. And there are a lot of computational works on those functions already because they are in some extent easier to simulate. For those higher level functions, they tend to involve a large scale brain networks. The many brain regions are involved, and hard to define the boundary of those functions. So there are far less work on these high level functions, for example, memory and consciousness. But this is exactly the topic we want to focus today. We want to perform some simulation about memory and consciousness. So first, I like to present my own work on memory, and then my colleagues Leon and Valerie are going to present their works on consciousness. So first, we need to define the boundary of the brain function, for example, like memory, because obviously it's also a very complex brain function by itself. We need to define exactly what we want to simulate. So fortunately, all those pioneers in neuroscience and psychology, they have developed a lot of tests or tasks to isolate the elements of memory that we want to study. So first, this slide shows a test called delay matched sample task. It's a task designed for studying working memory, a type of short-term memory. So in this test, the subjects will see a sample image at the very beginning, for example, like 1 second, and then the image will disappear, and then there will be a delay period, generally 1 second, sometimes 2 seconds or even longer. And then another image will show up on the screen, and the subject need to tell whether this one matches the sample or not. So this is a very simple task to test the subject's working memory. But by varying the complexity of the sample images and the length of delay period, we can already test a great number of hypotheses, like how long can we hold the image of the item in our working memory, and how complexity can our working memory be. And then this is another example of Sternberg's memory task. This is also a working memory task. In this task, at the very beginning, there will be multiple letters or multiple images that need to be remembered by the subject. And then, similar as before, there will be a delay period, or in Sternberg's test, sometimes is referred as retention period. And then one image or one letter will show up again on the screen, and the subject need to tell whether this one was shown among the set of items that he saw before. So this one is designed to isolate um, the capacity of our working memory. So how many items can we remember, like can we put into our short-term memory storage at one time? Similarly, there's another test, which is called n-back test. So the number n can be varied from 1 to whatever. Generally, I think the maximum is 5. So a sequence of images or sequence of letters will show up on the screen. And so this example is a 4-back test. So after 4 images, the subject need to tell whether the new image is the same as the one who appears like 4 times in the back. This is also a test to study the capacity of working memory. And then we have another example of long-term memory. So associative memory is the building block of our long-term memory. So we always associate things together. For example, when we see a tree, we have the image of the tree and the word of the tree, TREE, and the pronunciation of the tree. So we associate all those elements together, and that is called associative memory. So we can actually test a person's ability to build and store new associative memory. So like in this example here, some words are associated randomly, and three pairs are asked to be remembered by the subject. And after a long delay, because this is actually a long-term memory, so generally the delay can be, like several hours or even one day. And then the subject need to answer whether this is a pair of the other or not. Okay, now let's summarize a little bit what I just talked about. So how to create a mechanistic model for a specific brain function. So first, we may have some daily activity or daily phenomena that you feel interested into. So for example, like in our example, it's simply looking at a chair and read about the chair. And then we need to dissect this daily activity or phenomena into isolated brain functions. For example, in this case, visual processing, auditory processing, working memory, and long-term memory. And then we need to define the exact boundary, what we want to study about these functions. Like for example, here we want to study about memory. If our interest is about short-term working memory, then we can use delay matched sample test. If our interest is about long-term associative memory, and then we can utilize paired associative test. Once we have the specific research question, we need to design the pipeline of this study. So first, we need to have data because we are in the field of neuroscience. We always need data. So we can have fMRI, EEG, ECOG, and sometimes we can have animal recordings as well because they have better resolution. And then with this data, the first thing we can do is connectivity-based analysis and modeling. I guess you must have already seen a lot of studies in our group utilizing the virtual brain connective model. In this kind of analysis or modeling, we can determine the brain regions constituting the functions that we are interested in, and we can form some hypothesis already at this step. So which brain region is the critical hub mediating the function, or which connectivity is crucial for mediating the brain function. And then we can create a mechanistic model to reproduce the phenomena of brain function. And generally, these type of modeling tend to be multi-scale because we need to go into some deeper details. For example, like what are those single neurons doing while performing these brain functions? Like are dendrites or axons involved? Or are different cell types related to mediate the brain functions? And then after building this model, we need to consider some more sophisticated issues. For example, by produce a brain function with a hypothetical mechanism, we only produce like this is the possible way like the brain functions. But we need to add some biologically constraints to make it plausible because there may be multiple candidates that can simultaneously perform this brain function. And then we need more evidence, either from experiments or from computational simulations that can rule out some of the alternatives. And then we can confidently say, okay, this is actually what the brain does. And also, emerged mechanisms during simulation generally tend to be superior than those mechanisms that are designed to perform. This is because when you design your model to perform a mechanism, because we're doing computational works, you can have a lot of ways to produce that function or that phenomena. But emerging mechanisms tend to be like on top of the designed mechanisms that can match the experiment data better. And here I'd like to use some of my own published works as case studies. So first, so here is a study I published many years ago about modeling visual working memory. So first of all, um, let's dissect this study using the pipeline I just discussed. So first of all, the functions I I am interested here are visual processing and working memory. And then the tasks I chose are delayed matched sample tasks and Sternberg's memory load task. If you still remember, these are two tasks that are designed for testing working memory or short-term memory. And then I collected some open source data from human fMRI and from monkey single neuron recordings. And then I analyzed the connectivity underlying visual working memory. So the necessary connectivity patterns are the circuitries are a ventral visual pathway and a hypothetical working memory circuitry. And the last one, the hypothetical working memory circuitry, is the mechanism I wanted to talk about in that paper. Okay, so delayed matched sample task again. So this was the task I simulated in that paper. So first, a very simple image will show up to the model for 2 seconds. And then after 4 seconds, a new image will show up, and the model needs to determine whether this one is a match or a non-match of the very first image. And the connectivity I want to investigate at the ventral visual pathway from visual cortices, V1, V2, V4, all the way to prefrontal cortex. You don't need to remember all these names of brain regions. You just need to remember they are all involved in the visual processing and working memory. And then I embedded this visual ventral pathway and the working memory circuits into the virtual brain connectome. So on the background, all those gray dots represent a node in the TVB software package. But I isolated all these nodes that I was interested into further investigation and expanded those nodes into multi-scale models. And then I collected experiment data from some past examples from monkey single neuron recordings and human fMRI. So these experiment data are related to those nodes I just showed you. For example, the D1, D2 are the working memory nodes, and the IT are visual processing node. And these data are from the delay matched sample task performed by monkeys. And this is the simulated data from my own model. So you can see the D1, D2 simulation, the data generally matched the experiment data I showed here. And then the response unit R here responded to the second image. this is because like the input I gave here, the first input and the second input, they matched each other. So the response module of the simulation need to say, okay, this is a correct match. So in this task, we discussed how these memory units interact with each other, and this is the emerged mechanism out of the simulation. So we have two types of um, memory units, D1, D2. So D2 hold the very first image throughout the whole period, and then um, it will relate the second image with the first image. D1 units are a type of units who didn't respond to the first image, but it will respond to the second image and compare it with the first one. Okay, so here is case study number 2. Uh, this is also one paper I published 4 years ago, which is built on top of my first study. So the first study is about visual working memory, and here I built the auditory processing into the model as well. So this time it's a visual auditory bimodal working memory. So the functions I want to study this time is visual processing, auditory processing, and working memory. And the tasks are the same, delay match the sample task and Sternberg's task. And the data I collected from the field are still the same, human fMRI and monkey single neuron recordings. And this time, the circuitry I want to investigate is our ventral visual pathway and auditory pathway, and also hypothetical working memory circuitry, and also how visual working memory circuitry interact with the auditory working memory circuitry. Okay, so this time the circuitry obviously is uh, a doubly 2 times complex than the visual processing pathway. So because I have a auditory pathway in parallel and how the 2 pathways interact. And again, I embed the 2 pathways into the TVB software package. And here are the tasks I designed for the simulation. So on the left, I ran auditory delay matched sample tasks and the visual delay matched sample trials simultaneously. So for the auditory one, the model will listen to some sound, and then after delay, another sound will be played to the model. The model need to tell whether it's a match to the first sound or not. And then the model will see an image, and then after delay, another image. The model will need to answer whether the 2 images are the same or not. And then to make things more complex, I added distractors to delay match the sample task. So on the right is an example. So for example, I'm showing some visual stimuli, some images to the model. And during the delay period, I play auditory distractors to the model. So the model still need to answer whether the images are the same or not. But in the meantime, it need to process the auditory distractor. Although it's not important, uh, but if the sound is very loud, we found that the model will forget the visual images it just saw. So here are the simulation results. I just want to briefly show you. So on the left, it's a very simple auditory delay match to sample task. So same as the visual delay match to sample task I just showed you to you. So the model can correctly respond to the second image if it's a match. And then similarly, the model emerged two types of memory units, like D1 and D2 on the bottom, the second bottom and the third bottom. Uh, the D2 will remember the first sound throughout the whole test, and D1 only respond during the delay period and the second sound. And then on the right is the DMS, a delayed match to sample task with distractors. So there will be multiple distractors shown between the first image and the last image, and the model need to process the images and also the distractors. But at the final stage, the model still answer the question correctly. But there are some very interesting dynamics emerged in the simulation about the processing of distractors, just like I just said. If the distractors are very prominent, it will interfere with the memory that it hold in his work in the working memory circuits. This is very realistic, but actually um, we never know the dynamics underlying this, like how distractors make us forgetting things. And this is the main conclusion of this study. Yeah, here I just want to show you briefly how, like an example, how the model remembers different things. So on the bottom panel, the subfigure B, it's the visualization of a working memory space. So with the progress of the test, more and more items will be stored in the working memory space. Okay, so here is the final case study I want to present today. This is a ongoing project. It's almost done, but it's not published yet. So it's built on top of the first two studies. It's about modeling the visual paired associative memory. So the functions this time I feel interested in are visual processing, working memory, and the long-term paired associative memory. Just a reminder that the paired associative memory are the building blocks of our long-term memory and language. Like, for example, I saw the word memory. I know the pronunciation of the memory, and I know the concept of memory. So by reading the word memory, my brain associated a lot of elements together that are stored in my long-term memory already. So this process is called paired associative memory. So the tasks I want to test, I want to simulate are delay matched sample tasks and paired associative tasks. And the data, again, are human fMRI and monkey single neuron recordings. And the circuitries I want to investigate are ventral virial pathway and hypothetical long-term associative memory. So here is the model structure. Because this is way too complex, I only show each layer as one block, as one node. So I have this visual recognition system, which is basically the same as the first two case studies. So this visual recognition system is representing the human visual cortices that can process visual images. And then I still have the working memory system that I built in the first two case studies. So they can store the images passed through the visual processing system. And then the visual working memory will interact with a long-term associative memory system. It can retrieve items that are stored there. It can encode new items into the long-term memory as well. Here are the tasks I simulated with the model. So first, I still ran the delayed matched sample task. So first I show the model a cat, and then I show a cat and dog. The model needs to choose which one is the first image it just saw. And then about the long-term memory part, I trained the model to remember the number of random pairs. For example, like apples are associated with books, bananas are associated with chairs. And then during the simulated test, if I show an apple to the model need to answer, okay, if, if the book is a corrected pair of the apple or not. And here are the results. So in the long-term memory circuitry, two types of neurons emerged out of the numerous training loops and simulated tasks. So first, I observed some encoding neurons. They respond to the first item and its pairs. So basically, these neurons they respond to both items of the pair. And then I realized there are another type of neurons that they only respond to the second image if it's a correct one. So obviously these type of neurons, they are responsible for the retrieval of the paired memory. And then I found there are some hub units emerged out of the simulation. So in the subpanel A, following the arrow, you see there are only 4% of neurons, but they are involved in the encoding and retrieval of almost 90% of all the pairs. And then the other neurons, they are more or less involved in some pairs, but these 4% neurons, they are everywhere. They are almost active all the time. So I call them hub units. This finding matches the experimental literature that in our brain, some units are hub memory units that are involved in a lot of different encoding of the different types of memory. But surprisingly, I found that if I cut out this hub unit, it barely impairs the performance. So in panel C, you can see if I silence all those hubs, the model obviously performed worse than original, but still the correct rate, the accuracy rate is still amazingly high. This suggests that the memory circuitry is very robust and resilient. By simply cutting out those hub units, it didn't impair our memory that much. Yeah, and this is the main conclusion that emerged out of the simulation of long-term memory. And thank you. And this is the presentation of my work. And I can answer, if you have any questions, I can answer them at the end of the presentation. Thank you.
All right, thanks, Tim. Hi, everyone. Again, my name is Valery Bragin. We work together actually with Qin and Leon, and I will present also projects that we're doing together. But first, I just wanted to give a brief introduction into the subject of scientific study of consciousness. And let me try to share my screen. 1 second. Oh, can you see it now? In a full screen already, right? And also I wanted to mention that probably I might have a not very stable internet connection. So if something hangs or interrupts, shut out, let me know. I will just switch off my camera. Okay. So now we go into next topic, consciousness. So scientific study of consciousness has really matured over the last few decades, and it is no longer something hand wavy, something that people frown upon. Now it's kind of a real science. But when we say consciousness science, I wanted to highlight that two things that we now take for granted, but that would have sounded quite strange some time ago. First, we study something that still has no single strict definition, right? But the community agreed that, okay, green light, let's just go forward and we'll shape the subject as we go. And mm, Actually, what helps is that consciousness can be actually decomposed into different aspects. And mm, One basic split is between the state of consciousness and the content of consciousness. Uh, for mm, for content, for conscious experience itself, people often say this classical what is like to be a something, right? What is like to be human? What is like to be a bat? Paraphrasing Thomas Nagel here. Or what is like to be a chair, for example? Probably nothing, and that's one way to define consciousness negatively. Right? An object possesses no consciousness if there is no such thing as to be this object, to be a chair, right? Um, on the other hand, state of consciousness maybe is in some ways more tangible, and then can still be decomposed and even in two axes, along two axes. So we identify a level of awareness and the level of arousal or wakefulness. And along those axes, we can evaluate a given state, whether it's physiological state like sleep, wakefulness, drowsiness, or it's pharmacological like anesthesia, or it's pathological like disorders of consciousness of varying severity, of varying depths of impairment. So the first point I wanted to make here is that so yes, we don't have one strict definition of this of subject of consciousness, and some critics will say that it is not scientific enough, right? But we do have a kind of a fabric or structure around the around how we define consciousness, how we think about it strictly and scientifically, and it evolves, inspires new research, and that's kind of feeds back in a somewhat circular way. So we can go with it. And the second point, it was really revolutionary at that time, is that the field also agreed to rely, at least in part, on subjective experience, so, on subjective report of subjective experience of people under investigation, right, the subjects or patients. Uh, so not in a very aspect of every experiment, but in the study of both consciousness and perception, kind of very often we have to rely on this subjective report. Do I feel the stimulation or not? If it wasn't like near threshold stimulation, or can I see face or vase like here? And Sorry, lost it. Yeah. So if you think about it, it maybe even can sound kind of fine scientific to rely on something we cannot directly measure from the outside, and that we maybe cannot fully verify. So maybe there is, some conspiracy of subjects and patients all agreeing to intentionally undermine science. Um, but yeah, it's half joke, but half truth in methodological sense, right? But also, some experimental designs are report-free, and that's important that they rely only on this objectively measured activity. So it's apart from being more scientific in terms of what I've mentioned above, it also kind of makes results cleaner and separates the results from the artifact, from potential artifact of reporting activity. All right, it's even if they don't kind of have this evil plans of misleading us, still the record activity would be in this case more clear, and it's one of the case of debate between different theories of consciousness that I will mention a bit later. Theories of consciousness, there are a lot of them, as you can see. So yes, all this I mentioned, this hasn't stopped the researchers from studying consciousness both empirically and theoretically. Let me just maybe briefly mention some of the theories in more detail. It's not the main subject of this talk, and we don't really have time to go deeper. But I want just to name some theories, or better to say maybe groups of theories. Yes, it can be grouped in kind of classes. And yes, this is from Set and Bain review from 2022. And they identify this four major groups of theories of consciousness: higher order theories, global workspace theories, integrated information theory, and this re-entry and predictive processing theories. Global neuronal workspace theory, it's GNWT. It stems from earlier work of Bernard Baar's global workspace theory. So it was not neuroscientific, but general. And the metaphor Baar's used is a theater, actually, for his view. So it's like many specialized processes work in the background in parallel, like something was going on behind the scene, but only a small kind of maybe small amount of information of activity gets broadcast to a wider audience, like the stage in theater, and it becomes globally available and thus consciously perceivable. And later, neuroscientists Stanislas Dehaene and Jean-Pierre Changeux, they extended it and added a neuronal implementation to this idea. So that's how it's turned from global workspace theory to global neuronal workspace theory. Right. And so they key claim is that conscious access happens when information is amplified and broadcasted across this long range cortical network, especially like frontoparietal areas. So it becomes available to many downstream systems like memory, planning, language, action, as kind of other components of the brain activity. And also important predicted phenomenon that they have here is a so-called ignition. It's a like sudden and non-linear burst of activity of this coordinated activity, often also with strong frontoparietal involvement, and this occurs when a stimulus crosses a threshold for conscious report or conscious perception. And below this threshold, processing may still occur, but more locally and more transiently, so it doesn't result in the ignition. So in this global neural workspace theory, consciousness is not just this local processing in one area, but it's about global availability and global integration across this workspace. Another one, quite established and famous one, is the integrated information theory (IIT) by Giulio Tononi and Christoph Koch, I guess. And here it starts from a different approach. It starts closer to phenomenology, like what properties must any experience have, right, in principle. So they came up with so-called postulates about experience, like its intrinsic existence, composition, finiteness of experience, its exclusion. So something is not always there is something that you're not conscious about. And these postulates are meant to translate into requirements for the physical substrate. And the theory then kind of gives the mathematical quantity phi, and it is supposed to measure how much irreducible cause-effect power on itself a system has as a whole, kind of above its parts. So irreducible is a key word here because it's like tested by successively removing some intrinsic links within the system, or maybe perturbating them, and measuring how it affects this cause-effect power. And roughly, the more integrated the system is, the more recursive it is, the higher the phi, so the more consciousness it is, at least of course, in IIT's framework. And they claim, by the way, that if I understand correctly, that any physical system with certain neuroscientific angle, anatomically, IIT has often like emphasized the so-called posterior hot zone, like on the back of the brain, like regions like posterior cortex, where this sensory processing may occur and may be actually integrated. So that contrasted with the theories that put more weight on the frontal or frontoparietal broadcasting. Actually, there is a famous debate, frontal versus posterior kind of accounts of consciousness, and there is this adversarial collaboration, and it kind of didn't really settle the debate, I would say. So it perhaps favored posterior side a bit more, but both sides found a certain support, and both were actually challenged in their places. So I think the debate is ongoing, right? Another one that I wanted to mention is dendritic integration theory, DIT. It's actually it comes partially from colleagues here in Berlin at Humboldt University, Matthew Larcom and his colleagues. And very roughly, it proposes that layer 5 pyramidal neurons in cortex integrate feed-forward and feedback inputs, so bottom-up and top-down inputs across cortical layers. And yeah, we'll come back to it when we talk about our modeling work in a little more detail. Now, so even with all those caveats, we don't say that we are studying consciousness per se directly. So what we can model are so-called neural correlates of consciousness, NCC. Formally, this NCC, it's a kind of NCC of conscious state, this minimal set of neural events that together sufficient for that state. So in principle, the search for this NCC kind of it involved seeking these brain states and processes that are most closely related to consciousness, right? So it's kind of a correlation between processes and conscious experience. So, for instance, there are different NCCs that I think we'll come back to some of them later in this talk when we'll match the when this correlates, we'll match the experimental or modeling work that we'll discuss. And we also can't really avoid this distinction between the hard problem and the easy problems of consciousness. This was famously coined by philosopher David Chalmers in 1990s. So the easy problems, I think they're easy. Easy here is kind of relative, of course. They're about explaining these cognitive functions with their neuronal basis, right, and correlating them to this neuronal activity. This might be not easy scientifically, but why it's called easy problem because it's in principle they look like a standard neuroscience problem. And on the other hand, the hard problem is different. It asks why and how should any of these physical processes really give rise to the subjective experience at all. I mean, and the kind of the perceived quality of conscious experience. They actually call it quality. And I think I'm back, right? Okay, let me share again. Uh, I have been absent for a long time. I hope no. Uh,
No, not too much.
Just some
okay,
second.
okay, good. So I can probably maybe repeat. Can you see now my screen? My slides?
Yes.
Good. So probably I can repeat about about the hard problem. So hard problem is second. Hard problem kind of deals with how and why should any physical process give rise to exact conscious experience of something, right? And actually, how we can, from physical process, we can have this subjective feeling of qualitative experience, so-called quale, and on this what is like to be something or what is like to feel a certain experience. Yeah, in here we have this explanatory gap between objective mechanism and subjective internal experience. And I think classically NCC, neural correlative consciousness, this research sits mostly on the side of the easy problem, right? Or at least on the side of mechanisms we can study with experiments and models. Although some theories claim to speak more directly to the hard problem also. For example, IIT and certain versions of higher order theory, I believe they kind of claims that they trying to solve hard problem. But yeah, here in this talk, I think in our work we're more on the side of easy problem, of course. So and finally, before I hand it over to Leon, I just wanted to outline what we don't cover here. So we leave aside this theory of kind of roughly quantum consciousness, right? Is so-called orchestrated objective reduction, or by Roger Penrose, physicist, and Stuart Hameroff. And according to it, conscious moments may be linked to a hypothesized quantum process in microtubules that are related to reduction of wave function. So this is I personally find it interesting, but it's out of scope of the type of science we do in this field. We also completely leave aside machine consciousness and AI. Again, we're not dismissing it, but just not the scope of our work and of this talk. However, so yeah, we deal with human consciousness and actually with animal consciousness is included, and actually animal consciousness. It's actually we assume it not only exists, but also it may serve as a model for studying human consciousness. So we postulate some qualitative similarity among humans and other species. I mean, this is kind of practical approach, so we can use animal models and animal experiments to inform our work, which is, of course, in the end has a goal to understand human consciousness. Yeah, and with that, let me hand it to Leon. We'll go to more like substantial part, and then I will be back with also with some other models.
Okay, let me set up. Can you see my screen?
Not yet.
Not yet?
No.
No.
Yeah.
Yeah, now it's good.
Perfect. All right, let me quickly introduce myself before I start. I'm Leon. I'm a PhD student in the brain simulation section research group, and I'm working on modeling thalamocortical interactions during loss of consciousness in anesthesia. But before I tell you about this and also other modeling approach, computational modeling approaches, I will pick up from where Valery left us, which is kind of a theoretical overview of where we stand in terms of consciousness, and tell you a little bit about some empirical measures and what we like some findings. Also take this with a grain of salt, because as Valery already mentioned, it's more about NCCs than actual consciousness, of course. Okay. Wait a sec. All right. So you've seen this slide before in Valery's talk. I want to use it to show kind of that consciousness is not a a one-dimensional construct, so it's more complex, and there exist several spaces in this, or spots in this complex consciousness space, if we want, where where a subject is aware but cannot really show it through behavior, and that makes it also like a practical problem, not only a theoretically interesting topic, because we cross paths with these cases a lot in clinical practice. For example, there exist different types of coma where people are to a certain degree conscious, but cannot show that behaviorally, or a broader use case, let's say, in anesthesia, where we, of course, want to know if people are unconscious before the procedure starts. So we cannot only rely on behavior to tell if someone's conscious or not. We need a measure of consciousness, or a proxy for consciousness that optimally is derived from brain activity directly. And I will now talk about some exemplary cases. Of course, there is a lot of literature, but this will be out of scope again. So one prominent one, and also quite elegant one, that I chose here, is derived from the integrated information theory that Valery told you about earlier. The main takeaway of IIT is, or the guiding principle is, that consciousness needs a balance of two things, right? Integration and differentiation, if you want. And there is this measure, perturbational complexity index, that was developed by a research group in Milan, that kind of reproduced a proxy, or that produced a proxy for this phi, which is the measure for consciousness according to IIT, which is not practical to compute in reality, let's say. Starts with a TMSEG setup, where you apply a stimulus to one region and then record the evoked activity. And theoretically, in a case where there is maximal segregation, this stimulus would not propagate because regions don't interact. And in the case of maximal synchronization, the stimulus would maximally travel through the system and basically cause the same reaction in every region, which kind of makes this redundant. And according to IIT, the optimal spot would be somewhere in the middle there. And from this, they derived a measure that that kind of distinguishes different states of consciousness. This is the same concept in a little more in detail. We have a time series here, and a TMS stimulus that's applied at some point, and then we see the stimulus propagating or affecting several regions. And then you can quantify how much basically the system was affected by comparing if the signal crosses a certain threshold compared to the baseline. And then there are some mathematical steps involved that I will not go into detail here, that leave you with one measure that was actually shown in a validation study that could actually distinguish quite well between wakefulness and unconsciousness caused by several different anesthetics and sleep and so on. Okay. Whoops, sorry. Okay. We also observed these signatures of conscious states at other levels. One very prominent one would be the functional networks that we know from resting state recordings. And during loss of consciousness, we observe the reduction of these long-range connectivity networks, especially in between frontal and parietal regions. Sorry. And we observe this across different modalities, imaging modalities. And as a side note, just as perturbational complexity was kind of derived from IIT, these functional network breakdowns would correspond to other theories of consciousness, or most prominently, I would say the global neuronal workspace theory, where the idea is that consciousness depends on broadcasting information widely, which is disrupted during loss of consciousness, and we can see that through this measure. Okay. Also interestingly, this frontoparietal breakdown is a very widely reproduced finding in the whole field, also seen across different anesthetics like propofol, sevoflurane, and ketamine, that have different molecular mechanisms on how this state of anesthesia is reached. Okay. Speaking of anesthesia, I would now like to make the case that anesthesia is a good entry point or a good model to study loss of consciousness for us computational neuroscientists. The first one would be that it's in a clinical settings and it genuinely matters that we actually know if someone is unconscious or not. But also it usually gives a good or we get good data from these setups because there is a control of the conscious state, usually several conditions with different depths of consciousness, and it's reversible. And I would argue that this is also very important. We have a good entry point for modeling, which is the molecular mechanism. That is, I guess, known for the most anesthetics. All right. So that leaves me here, which is I spoke about the network level changes, the neural correlates of consciousness, and the molecular mechanisms that we know for most anesthetics, but mostly there is a mechanistic gap between those two levels that kind of differ in several orders of magnitude, if you want, in terms of level of resolution. And here is a good entry point for us to maybe try to get a connection between these two levels using a model. And I will now tell you about past approaches and my current approach on how I'm planning to do that. All right. But before I do that, let me tell you about a region called the thalamus. You might have seen it pop up here and there already in past slides. For example, in Valery's slide on the dendritic integration theory. So the thalamus is a hub region containing several different nuclei that you can segregate based on different criteria. And functionally, it serves different purposes. So for one, it serves as a relay to bring sensory information from, for example, the eyes or the ears or whatever to the cortex. But on the other hand, it also has reciprocal interactions with the cortex. For example, it enables cortical interactions, which is interactions between regions that kind of send signals through the thalamus back to another region. And it's also involved in the ARAS system, the arousal system, which makes us wake up, basically. Or, for example, so it has quite an important role in shaping also cortical dynamics. There are several ways to classify or to classify the nuclei. I'm gonna tell you about the most classical one. It's by projection type, and there's a rough. You can kind of roughly categorize two nuclei types. On the left side, you see what we call non-specific nuclei, and they are characterized by diffuse projections to the cortex. And on the right side, the blue nuclei, you see specific nuclei, and they're characterized by more or less direct, very targeted projections to the cortex. On the right side here, you see an abstraction and and my poor attempt to kind of graphically show these two types of nuclei and their connections. For the sake of simplicity, I will call the specific thalamocortical projections from now on type A, and the non-specific diffuse projections type B. Okay. As I talked about anesthesia earlier, there's a finding that these two different projection types are affected differently during propofol anesthesia. More specifically, the specific type A thalamocortical connectivity was not as reduced as the non-specific nuclei. You can see that burned down in this bar plot here. So we see the type B functional connectivity between thalamus and cortex was heavily reduced, while the other one not as much. And this leads me to this slide, which is that the non-specific group, so this side, the type B connectivity, is in the literature tied a lot to the state of consciousness and regulating the state of consciousness. But how that actually works, or how like the underlying mechanism is not really clear. There's a prominent, or there are two prominent fields. Actually, there are more, but you can roughly separate it into bottom-up versus top-down approaches or theories, where the bottom-up view is that anesthetics suppress this non-specific thalamus, or the nuclei, the non-specific nuclei, and those there are downregulated. So the cortex is then as a consequence also downregulated, or the cortical interactions. And then there's the top-down view, where it's basically the reverse, which where cortical communication breaks down first, and then the thalamic change in activity is a readout of this of this activity change in the cortex. And there is evidence for both theories, which keeps this discussion open. And this is a good entry point for the modeling community, because there you can kind of create a virtual experiment where you just test out or where you can create different conditions where you test these two theoretical mechanisms. Okay. Before I go into what I'm doing in detail, I will tell you a little bit about two modeling approaches that that are kind of within the same in the same topic. First, there's this paper by Lupi et al. Where they implemented, instead of homogeneous inhibition to the cortex, not to the cortex, to the whole brain, they implemented heterogeneous inhibition of cortical regions based on PET data of basically region-specific receptor density. And they found that this kind of increase in detail, so the heterogeneous inhibition of the cortex, mattered or improved their model and got them closer to reproducing the data, the empirical data they compared it with. And then there's this other paper by Miller et al., where they implemented propofol uniformly, but then implemented a virtual stimulus to the non-specific thalamic nuclei and could recreate some of the cortical dynamics and information theoretical properties of the wakeful state. Okay, so the takeaway here is that we know that the heterogeneity in the cortex has a relevant or is a relevant mechanism or has a relevant effect, and the importance of the matrix thalamus or the non-specific thalamus. But what has not been tested is the kind of maybe a connecting piece, which is whether what if there's a differential if there's a differential mechanism in the thalamus. And this is where I come in, basically, because there is a mechanistic hypothesis that in the thalamus there's actually not uniform inhibition, but heterogeneous inhibition. And one of the mechanistic hypotheses for that is that the specific or type A nuclei receive mainly excitatory input by, for example, the sensory organs, while the type B non-specific nuclei are also in circuits with cerebella and others, and subcortical basal ganglia circuits that also send inhibitory signaling to those nuclei. So when propofol increases GABAergic inhibition in the brain, there's a case to be made that these nuclei actually are more inhibited. The type B nuclei are more inhibited than type A. And what my aim with this with my modeling project is to see if we can maybe create a mechanistic link, if you want, or like a plausibility that maybe this mechanism in the thalamus is somehow responsible or linked to the network level connectivity reductions that I told you about earlier. Okay, so now to the modeling. I think I will not go too much into detail here because You probably heard about these kind of approaches a lot, but to keep it short, we created a structural connectome based on structural MRI data, where we have a kind of classical cortical parcellation, and then we connected to this cortical connectome a fine-grained thalamic parcellation. So we have now we have a connectome that represents the connections of a lot of nuclei to the cortex, and then we classified these nuclei according to this classification principles that I told you about earlier into non-specific and specific nuclei. Okay, so we now have this connectome with each region as a node. And in each node, we put a neural mass model. In my case, I'm using the so-called larger Break-Spear model, which symbolizes two neuron populations, and it has two inhibitory inhibitory parameters. The one represents basically a scaling of the inhibitory input to the excitatory population, and the other B is kind of serves as an analogy for the decay time of the inhibitory current. Okay, and yeah, basically bring these two things together, you get one of those brain network models that probably a lot of people have told you about. Okay, so the first thing we did here was to fit the model to resting state data. Basically comparing an empirical functional connectivity of the cortex to the best fit we could achieve. And then we set these global parameters fixed and continued and had kind of this virtual resting state. And then continued to implement inhibition to model propofol activity or the propofol mechanism of action. And we did this in two conditions. So one would be homogeneous inhibition, where it's kind of the null hypothesis that there is no difference or there is homogeneous inhibition across the whole brain, basically. Down here, you can see how we scale these parameters. So basically, you just linearly increase this one parameter, the inhibitory scaling parameter, and you prolong the inhibitory time constant. And then there's the heterogeneous condition where we implement the hypothesis that I told you about earlier, which is there's increased inhibition to the non-specific type B nuclei compared to the rest of the connectome. And this is how we implemented it here. So basically, there's this linear increase in the rest of the connectome, and then there is increase of inhibition in the type B nuclei. All right. And we did this in a 2D parameter scan. And what you can see here now is it's quite an abstract measure. So let me go step by step. On the top left, there is that's basically the resting state condition, so the default parameters. And then as we increase inhibition along these two axes, this measure, the measure that we are seeing here right now is thalamocortical functional connectivity. So thalamocortical functional connectivity type A specific and type B non-specific. And we see that as we go along inhibition, which is kind of to the bottom right, we get a decrease of functional connectivity between thalamus and cortex. And in the heterogeneous inhibition, it looks very similar, but there is kind of the spot here, starting at, let's say, yes, it's starting here, where you get this blue stripe in the bottom plot. And this so this bottom plot is a difference, simply the difference between the two top heat maps. And what this tells us is that here, the non-specific thalamocortical FC reduces more than the specific thalamocortical FC. And if we translate this into a bar plot, then we see that when we implement this heterogeneous inhibition, we get kind of to a similar ratio of functional connectivity reductions between thalamus and cortex, as we've seen in the empirical findings that I told you about earlier. Also, interestingly, or I think it's a cool finding, is that you can only get to this kind of spot by reducing both the inhibitory time constant and the inhibitory scaling, which is also close to what we see or what we know what propofol does to the inhibitory current. All right. With this, I will hand over to Valery, who will tell you about more fine-grained modeling of different states of consciousness.
Yes, thank you. Let me share again. Find the proper place. Right. So you can see it now, right? Or we call it part 2. That will be slightly different. I see a raised hand, or it just, oh no, it's gone.
Sorry, Valery, I thought that there is the end. Sorry.
Yeah. It's one more part, and we'll have all the questions in the end.
Okay.
Yeah, I hope you guys still kind of following. Or maybe we can have a 5 minutes break until 6 p.m. or it's fine. Sorry, I cannot see audience.
Yes, of
Oh no, okay, then yes, yeah. Let's then go on. It's fine. So Leon told you about what I would call the more widely used approach to modeling certain aspects of consciousness. So this whole brain neural mass, or sometimes neural field models. Some models are even phenomenological without a direct link to measurable neural activity. But on the other side of the spectrum, there have been also attempts to model specific mechanisms at more detailed at circuit level. So kind of here on the left, you can see quite intricate thalamocortical circuitry. Actually, this aspect is especially interesting at this level is this thalamocortical interaction, because it's believed to be not to rely on more specific mechanisms than those normally captured by coupling between neural mass models, hopefully. And also, the importance of thalamus really cannot be overstated, because in particular, stimulation of the central thalamus is one of the promising therapeutic approaches in patients with disorders of consciousness. It's a kind of a busy diagram, but you can see that deep brain stimulation or ultrasound stimulation applied to central thalamus, it's one of the main therapeutic targets. And actually, a central thalamus. This is a structure that it's quite small. It lives within this interlaminar group between this bigger parts of thalamus, and it has. I think Leon also mentioned this distinction. It has a diffuse pattern of innervation of multiple cortical areas. It's not even just the matrix thalamus, because I think matrix thalamus is a wider concept, and the central thalamus is even more specific, and it mostly targets deep cortical layers. It has specific laminar projection patterns, which also is believed to be important for its function, and we look into it in our models. And also, this is more instrumental for building computational models, is there is a consistent effect of stimulation of this also almost same thalamic areas in model organisms like rodents, and especially in non-human primates, like, for example, macaque. I think it's not easy, perhaps not ethical, to induce disorders of consciousness in macaques, like coma or minimally conscious state. But what people do instead is they anesthetize the animal, and they can apply, for example, deep brain stimulation, which can actually restore signs of arousal even while the anesthetic agent is still active. So, animal, for example, keeps breathing propofol, but it receives the stimulation and it gets awakened again. There are quite a few experimental studies on that, but I just wanted to highlight a couple of them. They both have the same experimental design. So they have anesthetized macaque and they apply deep brain stimulation. And in this study by Tasseri, they record functional MRI during unconscious, during normal states and unconscious state anesthetized, and then after bringing it back with stimulation to consciousness. And yeah, of course, we cannot tell for sure if macaque is conscious, so they use certain methods to evaluate, starting from open eyes and to some movement patterns and so on. But yeah, I think it's quite reliable. And another one is, again, same design, but they use transcranial recordings in some frontal and parietal areas. This is from Andre Bastos' group. Yeah, it gives us even more insight, even in laminar, because they can record from deep and from superficial cortical layers and from thalamus. Yeah, so it gives more insight into the mechanism of processes that's going on there. They actually show that the stimulation of the central lateral, I think in this case, CL nucleus, this part of the interlaminar thalamus that I shown, was most effective at restoring arousal and actually cortical dynamics under anesthesia, and the stimulation of some neighboring nuclei like meridorsal or some others, it was less effective. And they also used control sites outside the central thalamus, like the ventrolateral, which basically did not reproduce the effect. And this actually fits well with this clinical idea that interlaminar central thalamus matters especially for arousal. So not just any thalamic input. And also, one experimental finding that connects to this is a so-called anteriorization of alpha and more broadly beta power. So, I'm sorry. So they show actually in this experiment. So this is on the left. This is a recording from this Bastos work in macaque. They actually show that in unconscious state, against the general trend of decreasing alpha in unconscious, right? They observe that frontal alpha may actually increase, or at least become relatively stronger compared to posterior alpha. So this is called anteriorization, because it's kind of this alpha and beta power anteriorize, it goes to front. And this is one of this neural correlates of consciousness, NCCs, that we mentioned before. And it's quite well established in anesthesia. You can even see in this recording that how much anterior frontal zones have more alpha and the posterior have decrease in alpha. But it also has to do actually. Yeah, we can observe something similar in the disorders of consciousness here on the right. Not all of them types of disorders, but I think in non-anoxic, in traumatic brain injuries, we also can observe that relatively alpha power is more in unconscious state. And people have been looking into why this might happen, how alpha power can increase under anesthetic like propofol. And there are probably several models. I'll bring you one example. This is from Qin et al. From Nancy Coppell group. This is a spiky network model. It's where, so it's not a neural mass or like this mean field model, but here they model individual neurons, and they assemble it into populations with synaptic connectivity between cells and populations, and also between populations. And actually, it's also a type of modeling that we do that I will show you later. And the action of propofol is also modeled as in Leon's work, mainly as potentiation of GABA by increasing inhibitory strengths and the duration. I mean, the time scale of inhibition, so it becomes more prolonged. Each spike of inhibitory synapse will result in longer effect. And because of this, so here you can see that with more and more propofol applied anesthetic, cortical rhythm slowed down from this faster beta gamma towards alpha, and actually thalamocortical interaction also shift toward alpha with more propofol, and it's actually partially, maybe mainly, because of this inhibitory interaction within thalamus. So in thalamus, we have this thalamocortical projections that are excitatory cells that excite cortex. And also there is this thalamocortical nucleus, which is inhibitory, and it projects to thalamocortical projection nuclei, right? So it's kind of a shell around thalamus that inhibits the thalamus itself. And the key mechanism here is quite interesting. It's a current that is activated after a cell is hyperpolarized. So it's actually not excited, but when cell is inhibited, and after some time, it's kind of a rebound spike or rebound burst in thalamus. And also this, one of the currents in thalamocortical cells, T-type calcium, it's also it's sensitive to this kind of inhibition. And this kind of this pair of exciter inhibitor thalamus, they help to build up this alpha base. And the more propofol, the more this effect is there. So here, just showing this intrinsic ionic currents that have due to intrinsic time scales of this effect of these currents. It prefers alpha, and the thalamocortical alpha becomes more coherent with high propofol. And then yes, this actually quite early work, and I think it was extended more recently by others. And in our model, which I'll show, we also employ this kind of mechanisms. And also, probably even more prominent case on the circuit side, on the cellular and circuit side of modeling, is the role of this layer 5 cortical pyramidal cell. So this layer 5 cell is it's just called layer 5 because soma sits in layer 5, but really the cell is huge. It spans the whole depth of the cortex, and apical dendrites, the end of dendrite, are actually in layer 1, in the most superficial cortex. And the effect that was noticed a long time ago here is so-called backpropagation activated bursting. And its principle is that, so here we record, actually experimenters record from soma, from apical dendrite in red, and from somewhere in the middle. And if you just only stimulate the apical dendrite, you have this little bump in the apical current, in the apical membrane potential, which doesn't lead to anything. If you stimulate just soma, you will just have a single spike, a somatic spike, and that's it. But if you stimulate soma, actually, yeah, when you have somatic spike, you have this backpropagation action potential that goes up the dendrite. And if this coincides with the dendritic stimulation, with certain time window, right? You have this coincidence detection, and it decreases the threshold for burst in soma. So in soma, you can get this burst instead of a single spike. So this is kind of a nonlinear potentiation due to coincidence detection. And it's actually interesting. It's from also work from Matthew Larkum and Suzuki. And actually, it's in line with this broader picture of dendritic integration theory, the DIT that I mentioned. So it suggests that this L5 pyramidal layers can integrate bottom-up and top-down inputs across layers. So when the inputs arrive together, you can get this BAC, backpropagation activated firing, bursting, and you have this coupled streams of feedback and feedforward. But in the most interesting part is that was discovered several years ago is that under general anesthesia, as shown in the rodents, this coupling seems to break down. So when this animal was anesthetized, although you stimulate this apical dendrites, you don't have this coupling, don't have this bursting. So this is actually kind of top-down apical input no longer amplifies somatic firing the way it does in awake state. So feed-forward processing, actually, so bottom-up processing may continue to some extent, but this integrative mechanism of layer 5 cell is disrupted. And this actually is the mechanisms which DIT links to conscious processing. Yes, there are some other models that employ this little reduced model of layer 5 bursting, but I skip it in interest of time, and I think you already have a lot of information today. So I will go to our models that we develop here in the brain simulation section, and it actually builds on previous cortical macrocircuits and thalamocortical circuits from other groups. And but it brings in this L5 model with active dendrites, also not just full not this full morphological cell, right, but a reduced several compartment cell, but they still have geometry and that can yes, that can actually have this effect of bursting due to coincidence detection. Yeah, so we built upon the previous models that worked with this layer resolved cortical circuit. It has excitatory cells and two classes of inhibitory cells. And it actually was they had a frontoparietal to RS model. We'll have more RS. And they have more thalamocortical input, and they explored some attention experiments that deals with attention. So it's kind of partially overlaps with what we're interested in. And in our case, we have this microcircuit. It's still reduced, simplified, of course, but it's has superficial and deep layers, and this layer 5 cell that helps coordinating between the feedback and forward streams. And more cells are single compartment, so just the point neuron, point in space. But yeah, layer 5 neuron, as I said, is multi-compartment, which allows for this BAC firing. What is also important for this model is that it's not only cortex has laminar resolution, but cortical and thalamocortical connectivity also respects this depth of source and target populations. And it's based on tracked tracing studies, mostly I think in non-human primates. We reconstruct the different classes of thalamocortical projections corresponding to different thalamic nuclei. And as I mentioned before, this DBS efficacy, both in experiment with anesthesia and in patients in disorders, they highly dependent on nucleus type. So central thalamus, especially this cells like nuclei like centrolateral or centromedian parapascular, they're more effective in restoring arousal. And by contrast, some nucleus like metadorsal or pulvinar, they are central for other aspects of thalamocortical communication, but they are not the main targets with the goal I mean, not the main targets when goal is to bring back the arousal. It was this thalamocortical circuit. We did some preliminary simulation, so we tried to reproduce this effect of emergence of frontal alpha with propofol. So with increasing propofol factor, we have slowing down cortex and more strong alpha, and also this on the bottom thalamocortical coherence, because we have it's actually relies on also potentiating of thalamic alpha, as I described before. So we can observe that with more this is a power spectra of thalamic pair, or kind of two populations of projection cells and inhibitory reticular cells. And with more propofol, it gets more kind of more bound to alpha range, and is also following when we apply more and more high frequency stimulation, it still kind of, it doesn't follow it, but it keeps kind of stuck more or less on this alpha frequency. Yeah, and so this was a kind of, I was showing this single thalamocortical circuit, right? which was validated. Actually, it's in progress. We are validating against anesthesia, but in the end, the final goal is we're working toward extending this to the network level because we believe that phenomena we want to model are highly distributed and they cannot be captured by a local microcircuit alone. So in our model, we actually here, yeah, when we go to network scale, we're not using the whole brain model. It would be too expensive. We restrict ourselves to like several cortical areas that are considered to be from literature search and from theory considered to be more kind of relevant to consciousness state. And so it's mostly a higher order and associative ones. It's actually many of them are on the intersection of resting state networks like default mode network, frontoparietal, salience network, some of also attention network. And here actually we constrain the model with functional connectivity that from this macaque experiment with thalamic DBS that I've shown before from Tasseri. And the ultimate goal, it's absolutely work in progress, very preliminary results. And the ultimate goal is to bridge this pharmacological loss of consciousness, like anesthesia, which heavily depends on the pharmacology, right, on the GABAergic synapses, to bridge it to the structural damage in DOC, which has a structural level and network level phenomenon. So we believe that it can produce these overlapping effects, and eventually you can see kind of something useful about restoration of consciousness for patients and for with the therapy, based on insights from animal work. So this is where actually, although we cannot be sure that phenomenologically animal consciousness is the same as human consciousness, but we can use animal models as a kind of entry point and some help to develop human models to help constrain them and validate them. With that, I wanted to thank you. It was a long, a lot of information, long talk. I wanted to say thank to our supervisors and collaborators here in brain simulation section and also in other parts of the world. And I think we now have some time for questions regarding all the talks.